Welcome to Bio-Rad's workshop on CRISPR, cut, paste, and confirm, real CRISPR gene editing and PCR genotyping. My name is Leigh Brown, and I'm a curriculum and training specialist for the Explorer education team at Bio-Rad Labs. If you have any questions about this presentation or any of our other labs, please feel free to email explorer@biorad.com. I want to give you a little bit of background on Bio-Rad, just in case you're not familiar with our company. We're a biotech company based in the San Francisco Bay Area, started by a husband and wife team, David and Alice Schwartz, back in the early 50s. And now Bio-Rad makes materials and equipment for scientific researchers, pharmaceutical companies, and clinical labs all around the world. In the late 90s, one of the employees, a former high school teacher, wanted to bring biotechnology labs into the classroom. And so he founded the Bio-Rad Explorer program, packaging up biotech supplies and curriculum into kits that allowed teachers to do biotech in their classroom. Our Explorer team focuses on supporting classroom teachers by developing innovative labs and curriculum and supporting them through workshops like this one. We have lots of videos and other resources on our website at explorer.biorad.com. Let's talk a little bit about what we're going to cover in this workshop. If you're not already familiar with CRISPR-Cas9, we're not going to get too far into it in this particular workshop just because we won't have time to cover the lab itself. But you can always go to biorad.com/outoftheblue to find our CRISPR explained video, which is narrated, and it gets into a great overview of CRISPR applications, how Cas9 works, and also goes into more depth on how to use the CRISPR paper model that I'll touch on in this workshop. For this video, I'm going to assume that you understand bacterial transformation, but if you need a refresher on that, you can go to biorad.com/classroom resources, scroll down to where you see the YouTube video links, and you can find all the biotechnology videos linked there. The bacterial transformation one is a hands-on one that you can watch a teacher performing in the classroom, and also gives explanations on what you're seeing. So that's a good way to kind of get a refresher on bacterial transformation. In general, we recommend that students have already done some bacterial transformation before they go on to do this lab. This lab uses a bacterial transformation, but we already assume that they understand how they work and that they're somewhat knowledgeable on the process itself. So we really recommend that they do this after they've already done something like pGlow, for example. This might be a good capstone project. So in this workshop, we're really going to focus on that out-of-the-blue CRISPR gene editing lab, and then we'll go on and talk about the out-of-the-blue CRISPR genotyping extension lab. And finally, I'll show you some classroom resources that'll help you teach more about CRISPR in your classroom. This year, the Nobel Prize in Chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna for their discovery of CRISPR-Cas9. So with the out-of-the-blue CRISPR gene editing and genotyping extension labs, your students can actually perform that Nobel Prize-winning science right in the classroom and confirm that they use CRISPR to edit their bacterial genes. It's pretty exciting stuff. In this lab, students use CRISPR technology to edit the chromosome of E. coli. Let's start off by looking at something you're probably already familiar with. Normally, E. coli forms creamy white colonies that look like the ones that you see here. E. coli have a gene in their chromosome called LAC-Z. It codes for an enzyme called beta-galactosidase, or beta-gal. Beta-gal normally breaks down lactose. It can also break down a substrate called X-gal, which is colorless. When X-gal is broken down, it produces a blue color. So bacteria with a functional LAC-Z gene will be blue when they're grown on media that contains X-gal. On this plate, we see bacteria that are expressing LAC-Z, so the colonies are blue. Now, what happens if LAC-Z isn't functional? Bacteria without the LAC-Z gene do not make beta-gal, so they can't break down X-gal, and the blue pigment isn't made. When these bacteria are grown on plates with X-gal, the colonies are white. We can think of LAC-Z as a model for any other gene that scientists may want to modify. Scientists can modify genes by deleting, inserting, or replacing a sequence. By using CRISPR, they can target and edit specific genes. In this lab, students will start with the functional LAC-Z gene and then use CRISPR to cut and edit the DNA so that it's non-functional. Okay, time for a quick refresher. We often focus on how CRISPR technology is used to cut DNA, but CRISPR gene editing involves two broad steps: cut and repair. First, Cas9 and the guide RNA form a complex. Together, they find the target DNA based on the guiding nucleotide sequence of the RNA. The DNA unwinds, and the complementary sequence binds to the guide RNA. Then, Cas9 makes a double-stranded break, cutting the DNA. What happens when bacteria have a double-stranded break in their chromosome? If they don't repair the cut, it's lethal. So bacteria have evolved ways to fix breaks in their DNA so that they don't die. Now let's take a look at the repair process. Here we have our target DNA, which has been cut. Bacteria can use a process called homology-directed repair to fix a double-stranded break. In this process, donor DNA is patched into the cut. Bacterial repair machinery. The donor DNA has what are called homology arms, which are sequences that match parts of the destination DNA. The homology arms help the donor line up correctly. So, we need 4 components for CRISPR gene editing: Cas9, the guide RNA, donor DNA, and the repair machinery. Remember, in this lab we're starting off with E. coli that have a functioning LAC-Z gene. We'll use CRISPR-Cas9 to cut the DNA and then repair the break by inserting donor DNA. We've engineered this particular donor DNA with a stop codon. When this piece of DNA is inserted into the cut LAC-Z gene, translation will stop early and LAC-Z will no longer function correctly. We've created a Cas9 paper model that you can use with your students. You can download it for free from our classroom resources page at biorad.com/classroom resources. Students can cut out the Cas9 enzyme, target DNA sequences, and guide RNA. Then they can slide the target DNA along the Cas9 until they find a match with the guide RNA. Use their scissors to cut 3 base pairs from the PAM side. This is explained in more depth in the student manual. Now that we're done with the refreshers on CRISPR, let's talk a little bit about this particular lab protocol. The procedure takes a little over an hour, but there is a stopping point if you have shorter class periods. Each student group will do 4 different bacterial transformations, plate them on plates containing excal, and incubate them overnight. We've already mentioned the 4 components that you need to edit DNA: Cas9, a guide RNA, a donor DNA, and the bacterial repair machinery. Now, let's take a look at where each of those components fit into this lab. First of all, we need Cas9. In this lab, the E. coli that we're using have been engineered to continuously express Cas9. We also need the repair machinery proteins. We've put genes for the repair machinery proteins under the control of an arabinose promoter. The bacteria are grown on 2 types of starter plates, one with arabinose and one without arabinose. When starter bacteria are grown on media with arabinose, the repair machinery proteins are made. Without arabinose, the repair machinery is not made. We'll be using bacteria from each type of starter plate, so students can see what happens both with and without the repair machinery. We'll supply the bacteria with the other 2 components we need, the guide RNA and the donor DNA. That's part of the power of CRISPR. We get to provide the instructions for where to cut and how to repair. We're going to add the guide RNA and donor DNA by transforming our bacteria with 2 different plasmids, PD for plasmid donor and PDG for PLZDonorGuide. Both of these plasmids have the donor DNA sequence. PDG also has the DNA that encodes for the guide RNA. PD does not. So bacteria transformed with PD will not be able to make the guide RNA. Now let's take a look at what happens once we have all the components that we need. Remember, in order to edit the target LAC-Z gene, we will cut within LAC-Z and then repair that cut with the donor DNA. The bacteria from our starter plate are already expressing Cas9. Once we add the PDG plasmid, the bacteria will make the guide RNA. Now the Cas9 guide RNA complex can find our target LAC-Z DNA in the chromosome, unwind it, and make the cut. Let's take a look at the repair side. Here's our cut DNA. We get the donor DNA with the stop codon from our PDG plasmid. The bacteria have repair machinery proteins since they were grown on plates with arabinose. That repair machinery can patch the donor DNA into the cut and make the repair. That donor DNA has a stop codon, so once it's inserted into LAC-Z, LAC-Z no longer functions correctly. We just talked about what happens when we introduce all the components we need in order to edit genes with CRISPR-Cas9. But how do we know that we did CRISPR? We can control for some of the components we put in and then see what happens if we don't add them. In this lab, all the bacteria have Cas9, and we're adding donor DNA to all of the samples as well. We can control for the guide RNA by transforming with either the PD plasmid, which does not have the guide RNA, or the PDG plasmid, which has the DNA sequence for the guide RNA. And we can control for the repair machinery by transforming bacteria that were grown without arabinose, so the repair machinery is turned off, or with arabinose, so the repair machinery is turned on. Students will perform a total of 4 transformations, adding 1 of each plasmid to the bacteria from each type of starter plate. You might want to pause the video here and think about which processes are going on in each of these samples. Which samples will have the components needed to cut the target DNA? And in which samples will the DNA be repaired? Your students can fill out this information in the manual. Here are 3 of the phenotypes that you're likely to see in this lab. No You can use this slide and have students put in breakout rooms or lab groups in order to discuss which of the phenotypes they'd expect to see from each of these 4 tubes, A, B, C, and D. I usually just have them delete out the phenotypes that they don't expect to see. This takes a lot of thinking. It'll take them a good 10 or 15 minutes to kind of work through this in their mind. I'm going to just pause it here and let you make some predictions on what you think. You're welcome again to ask questions in the chat or talk to one another. You can always email me afterwards if you're still getting kind of stuck. It does take a little bit of time to think through this, but let me show you some of the results that we've gotten from other students. So here's what one of our groups came up with in a previous workshop. They decided they'd see blue colonies in sample A, no colonies in sample B, blue colonies in sample C, and white colonies in sample D. We can go over this later. I can talk about it in the chat, but these are your expected phenotypes from each of these plates. As an instructor, you need to pour the different plates for this lab. There's 3 different types. If you've done Piglo or something similar, it would be very similar prep to that with 1 additional type of plate. And you'll also need to streak out some starter colonies. Now let's get on to the student protocol. So your students will label 4 tubes A through D and put them on ice. It's a good idea to keep your tubes on ice as much as possible during this lab. They'll add 250 microliters of transformation solution to each tube. And then they'll use a loop to pick up 5 colonies from their X starter plate. That's the one without arabinose, and put those colonies into tube A. Swirl the loop for about a minute, make sure all the bacteria is off, and then repeat that for tube B. 5 more colonies of from the IX starter plate into tube B, and then put those tubes back on ice. They'll use a new loop to put 5 colonies from the IX era starter plate, that's the one with arabinose, into tubes C and D. And again, they'll swirl those loops for a while to make sure all the bacteria is off and put. Next, they'll use a pipette to add 10 microliters of the PLZDonor, which is labeled PD, into tube A. So if you remember that PD just has the DNA for the donor DNA, it does not have the DNA sequence to make the guide RNA. They'll put that into tube A and then use a new tip and put 10 microliters into tube C, flick it a few times, and then put it back on ice. Next, they'll use a pipette to add 10 microliters of the PLZDonorGuide, or PDG, into tubes B and D. And again, they'll want to switch tips between those tubes, flick the tubes to mix it up, and put it on ice. They'll incubate those on ice for 10 minutes. And just as a reminder, what we've got in those tubes is the transformation solution, calcium chloride. It has either the bacteria from the plate without arabinose or with arabinose, and it has either the plasmid with just the DNA donor, or the plasmid that has the DNA donor as well as the DNA sequence to make the guide RNA. Okay, so we have all 4 of those tubes on ice for 10 minutes. If your students have a stopping point here, there's an activity in the manual that they can do where they're comparing the specificity of Cas9 to restriction enzymes. So this allows them to use some math and data analysis to determine and see how much more specific that guide RNA is than something that they may already be familiar with, a restriction enzyme, which also cuts DNA. A guide RNA recognition sequence is typically 20 nucleotides, whereas a restriction enzyme recognition sequence is usually 6 nucleotides. So they can see how much more specific the guide RNA is and the CRISPR-Cas9 system is. After a 10-minute incubation, your students are going to then take their ice tray over to a water bath or dry bath set at 60 degrees and do a heat shock for exactly 50 seconds, and then put them right back on ice. It's real important that this step is pretty fast. They don't want to dilly-dally and walk around the room and let their tubes heat up slightly. They want to go straight from the ice bath to the water bath or dry bath and straight back into ice. If you are using a dry bath, I like to fill up the wells in the dry bath with water and let it heat up as well. That'll be a more efficient heat transfer into the tubes. Next, you'll add 250 microliters of LB to each tube, Lurea broth. And they can close the tubes, flick them to mix, and just let them sit on the lab bench for 20 minutes at least. Or if you have a shorter lab period, this is a good stopping point. It's okay to leave them overnight. After the bacteria have recovered a bit, you'll label 4 plates A, B, C, and D. These are all IX plates. They don't have arabinose because that arabinose process has already been taken care of from the starter place. They do have spectinomycin in them. Spectinomycin is going to select for the transformed colonies. And so the students will label these A, B, C, and D. They can add lab group and initials. They want to always label the bottom of the plate on the agar side around the edge. Next, they'll just plate their bacteria on these plates. So 100 microliters from each tube onto the appropriate plate. They can use a new loop each time, obviously, and they want to just swirl the bacteria over and cover as much of it as they can. They're not trying to streak for individual colonies here, just back and forth in several different directions so that they get a good coverage on there. Next, they'll just stack that plate of the stack of 4 plates up, tape them up, and incubate them at 37 degrees for at least 24 hours. If you don't have an incubator, you can incubate them at room temperature for a couple of days. They want to check for blue colonies after 2 days, and if you don't see any at that point, they can put them in the fridge for another 1 to 5 days. I typically see blue colonies on mine after 2 days though, but if it's maybe real faint blue or they're just not seeing, some of them aren't sure if they're seeing blue or if they look white, stick them in the fridge and see if that helps. At this point, they can count their colonies and just use a Sharpie to mark on the bottom of the plate. If they have a bunch of them, there's so many that they can't really count them effectively, they can divide that plate into quadrants and count the colonies just in one quadrant and then multiply by 4. Here are the 3 phenotypes that your students are likely to get from this lab. They could see a phenotype of no colonies, a lethal effect, white colonies, which would indicate that the bacteria had been gene edited, or blue colonies, which would indicate that the bacteria had not been edited. You can again pause the video here and think about which of these phenotypes would they be most likely to see with each of the 4 tubes. And next, we're going to go into how they can confirm that they actually edited their bacteria using CRISPR by using the Genotype Extension PCR kit. Let's go over a quick overview of the gene editing protocol that we just talked about and kind of put things in the big picture here. So if you recall, in the CRISPR gene editing lab, students used a simple bacterial transformation to edit the LAC-Z gene. They started off with blue bacteria with a functional LAC-Z gene, then they added plasmid DNA, which had the directions for where to cut and what to insert. In this case, it was a stop codon into LAC-Z. Next, they did a heat shock, plated their bacteria, and looked at their results a couple of days later. If the bacteria were successfully transformed and edited, the LAC-Z gene would no longer function and they'll form white colonies. In the genotyping extension, students will look at their bacteria from plates C and D. If you remember, plates C and D both came from bacteria that were grown in the presence of arabinose, so the repair machinery is turned on. The difference between these two is the bacteria on plate C were from a transformation where the plasmid only had the directions for the donor DNA. It did not have the directions to make the guide RNA. And on plate D, those bacteria had the plasmid with the donor DNA as well as the directions to make the guide RNA. Now, colony color is a visible phenotype that indicates whether the LAC-Z gene is functional or not. Students can't really visualize the DNA just by looking at their bacterial colonies, but they at least have some indirect evidence here about the state of the LAC-Z gene. If you were to look at these 2 plates, which plate would you say has a functional LAC-Z gene? Which plate likely has an edited or non-functional LAC-Z gene? And then what evidence could you use to support your claim? Whenever scientists edit genes, it's really essential for them to verify that the edit was made in the correct location and also that the edit was successful as it was intended to be. In the gene editing lab, the color of the colonies, the phenotype, gives us an indication that our edits were successful. But in order to verify that that edit was made in the correct location, we need to look at the genotype. Why is it so important that scientists check their work? Well, let's take a look at a real-world example. Normally, the horns of dairy calves are removed. Scientists at a startup company called Recombinetics used gene editing to swap out just a few nucleotides so the dairy cows never grew those pesky horns. But when the FDA took a closer look at the genome of the cows, they discovered some remnants of the cloning vector, including bacterial genes for antibiotic resistance. The company had previously claimed that the animals were 100% bovine, but they had neglected to verify their work by genotyping. Because the gene-edited calves had traces of that bacterial DNA, they were now considered genetically modified organisms, GMOs, and thus subject to FDA regulation. This story really drives home the importance of genotype verification to ensure your target DNA was edited in the way you intended and that there weren't any off-site targets. Here's a quick overview of the timing for the genotype extension. The exact timing is going to depend on the length of your classes. Once your students have completed the gene editing lab, they'll come up with 2 explanations for their results and think of what information they need to rule out one of those explanations. On the next lab day, they'll extract genomic DNA from their bacterial colonies. This can be a stopping point if you have shorter periods. If you have longer periods, they can go on and amplify their DNA using PCR to see if the LAC-Z gene was modified. The next day, they'll use gel electrophoresis to analyze their DNA. There's 2 visualization options that we'll talk about later on. Finally, they'll analyze the genotype of their bacteria and compile their evidence into claims about gene editing. In this lab, students are asked to use their results as evidence to support their claim about whether gene editing had occurred or not. They're then asked to come up with an alternative claim that could also support their observations. They can consult with other lab groups for other explanations, or they can use their knowledge of bacterial gene expression and cell biology to come up with an alternative claim. As a teacher, you might want to think about how you could guide students through their thinking here. In the genotyping extension, students extract the DNA from their bacterial colonies, use PCR to visualize the genotype, and determine if the DNA was modified as they expected. The genotyping extension uses 3 different primer sets in a multiplex PCR reaction. The first set is designed to detect unmodified LAC-Z. One primer in this set binds directly to the target site. If the site was modified, the primer won't bind. If the target site was not modified, then this primer set will yield an 1100 base pair amplicon. The second primer set is designed to detect edited LAC-Z. One primer in this set binds to the insert, and if the bacteria were successfully gene edited, PCR will yield a 650 base pair amplicon. The third set of primers will amplify an unrelated region of the bacterial chromosome, far outside of the LAC-Z gene, as a control. This allows students to verify that they successfully extracted and amplified bacterial chromosomal DNA. This primer set will yield a 350 base pair amplicon, whether or not LAC-Z was modified. Students can predict the sizes of amplicons they would expect to see from their starter plate, their control plate C, and their experimental plate D. If the target cut site was not modified, which amplicons do you think they'd expect to see? What if the target cut site was edited? Let's take a quick look at the student protocol for the genotyping extension. Students will be using 3 of the plates from their Out of the Blue CRISPR gene editing lab: a starter plate, plate C, and plate D. They'll label 5 screw cap tubes S, C, D1, D2, and D3. They can add their initials too. They'll give the instagene matrix a flick to resuspend it and add 250 microliters to each tube. Next, they'll use a pipette tube to pick up a single colony from the starter plate, putting it into the S tube. They'll use a new tip to pick up a single blue colony from plate C, put it into the C tube, and they'll use a pipette tip to pick up 3 unique white colonies from plate D and put them in 3 individual tubes. They need to, again, use a fresh tip each time. When they're adding the bacteria to the tubes, they want to swirl the tip around until there's no more bacteria remaining on the tip. They give the tubes a flick for a few seconds to mix them, and then put them in a dry bath or water bath set to 56 degrees for 15 minutes. They'll flick the tubes again to mix, and then move them to another dry bath or water bath, this one's set to 95 degrees for 8 minutes. Again, they'll flick the tubes for 10 seconds or so to mix, and at this point, the samples can be stored in the refrigerator if needed until the next lab period. If you have longer lab periods, you can just go ahead and start the PCR at this point. They need to spin down their tubes at 6,000 G for 5 minutes or 12,000 G for 2 minutes. This will spin the instagene to the bottom of the tube. Next, they'll label their PCR tubes and add 10 microliters of master mix plus primers to each one. Now, this is the part where they want to be very careful. So they want to add 10 microliters of supernatant, that's the liquid on top of the instagene, from each of those screw cap tubes into its matching PCR tube. They want to be real careful here that they don't stick the tip all the way in the bottom and accidentally pick up some of the instagene matrix beads. If they do, those beads will inhibit the PCR reaction. Once they've transferred their samples from their starter, from S, C, and D1, 2, and 3, they're going to use a new tip and add 10 microliters of positive PCR control DNA to the positive PCR tube, and 10 microliters of negative PCR control to the negative PCR tube. Cap the tubes, put them in the thermocycler, and run the cycle. You can leave the tubes in the thermocycler overnight, or they can transfer them into the refrigerator until the next lab period. The next day, they'll need to spin all their tubes down, add loading dye to each sample, and then run their samples in a 1% agarose gel. And you can see what they load here, 20 microliters of the molecular weight ruler, 15 microliters of all their samples, as well as the 2 controls. And they can run the gel according to your directions and stain as you want them to. Students can sketch out their gel electrophoresis results here and then decide if the results support their claims or not. You can see some sample results here. The molecular weight ruler is in lane 1. Lane 2 has the positive control DNA, which contains all 3 target DNA sequences. Lane 3 and 4 have amplified DNA from the starter plate in lane 3 and the control plate C in lane 4. These bacteria have unmodified DNA, so you can see the larger 1100 base pair amplicon, as well as that 350 base pair control amplicon. Lanes 5, 6, and 7 have amplified DNA from plate D, the experimental plate with the white colonies. All 3 of these lanes have the 350 base pair amplicon from the control DNA. Lanes 5 and 6 also have the 650 base pair amplicon. Remember that the primer set 2 binds to that modified LAC-Z, where that donor insert is, and makes the 650 base pair amplicon. Lane 7 only has a band for the control DNA. Have your students discuss scenarios that could have given this result. You're pretty unlikely to see this, actually. Lane 8 contains a negative control, so you can see those faint primer dimers down at the very bottom, but there's no amplified DNA band. There's a capstone project that's available online, again at www.biorad.com/classroom resources. Your students can look at different diseases, and then they can get into BLAST and look at the bioinformatics of designing their own guide RNA, determining where a good target site would be to bind that reduces the possibility of off-site targets as well. This really gets them a chance to get in the mind of a scientist and how they can design guide RNAs that are specific to identify specific targets in genes that we may want to modify. So it gives them an opportunity to do some research on the disease as well as get some bioinformatics experiences under their belt too. The teacher prep for the genotyping extension is pretty straightforward. You're just going to aliquot out the solutions, make the gels and the buffers. And we have all of our new kits now come with these quick start guides. These are really nice. As soon as you open the box, it's kind of like a printer, manual, so you don't have to dig out the whole manual and read through it right away. It gives you information about the timeline and the materials that are in the box, where to store things, if they need to be stored in the fridge or the freezer. It gives you space to write down where you've stored it, and also space to record the batch number in case you ever need to call tech support with any issues. There's then on the inside kind of an activity timeline that's graphical and gives you a good idea of what you're going to be doing with both the prep and the student portions of the lab. So here's what the example of where you should store all your components, what that looks like. The instructor guides for both of these are available online. We do not include the answers in the instructor manual anymore. They are included as a hard copy within the kit itself, but the manuals are only available online. So you're welcome to download those whenever you'd like. You can get them at biorad.com/out-of-the-blue for both the out-of-the-blue CRISPR gene editing kit and the genotyping extension. They're under the Documents tab. Here's what that instructor answer guide looks like. Again, we tried to remove the answers from the downloadable instructions just to kind of lower the chance that students might get their hands on it. The genotyping extension is available with or without the electrophoresis reagents, agarose buffer, and DNA stain. You can also choose to purchase it with the electrophoresis reagents and have your choice of either UView or FastBlast. UView is a loading dye and stain, and students can view their DNA using a UV lamp or transilluminator immediately after they run their gels. So this is really nice, it's quick, it saves a staining step, and it's not messy. FastBlast is a DNA stain that you've probably used before, or something similar, that requires staining for some period of time, generally 5 minutes or overnight. I prefer the overnight one just because it doesn't require destaining. FastBlast doesn't require any equipment to view it, but it is a bit less sensitive than UView, but you can just see it with your naked eye, which is nice. Here's the ordering information. Again, you can find the kits at biorad.com/outoftheblue. Manuals are available under that Documents tab, and look under the Ordering tab for information on all the different configurations. I've listed just a few of them here, but they're also available with or without the electrophoresis reagents, and you can also get the Genotyping Extension bundled with the Out of the Blue CRISPR Kit. So these are just a few of the iterations that we have. Typically, the kits will come with the consumables and the plastics that you need to do the lab. It's enough for 8 groups of students. We suggest that they work in lab groups that are 2 to 4 people, depending on the size of your class and the type of class that you're teaching this in. The refills are also available, and those refills just contain the reagents only. They do not contain any of the plastics. So that's the difference between the refill and the full kit. We have a lot of different CRISPR resources available online. We have YouTube playlists where we have videos that show you how to go through the labs. We have a great CRISPR Explain video that we've done, Damon Ty has done and narrated, and that'll give you more of the background of CRISPR. We also have playlists that we've put together that are just other people's CRISPR videos, the ones that we've used and enjoyed and that teachers have recommended. So we've gathered all those together in a playlist as well. We have infographics, and we have, again, remember that model, the bioinformatics activity. Those are available for free on our website. You can get to those, again, biorad.com/outoftheblue, or you can also go to the classroom resources page, which is biorad.com/classroom resources. If you're teaching a course where students need to be socially distanced or are attending school from home, we've also created resources for these labs and a lot of our other labs. You can find a list of the labs that are suitable for socially distanced and or at-home labs by going to www.biorad.com/adapt. And there you'll find a really detailed list of the labs, how many workstations it will serve, whether it's appropriate for in-class or at-home use, and how the adaptation might look. Again, this is more of a starting point. Teachers' labs are so individual, so this is meant to just give you kind of a guideline of how you might tweak the protocol and the prep in order to get more lab stations per kit. We have detailed directions that are in a PowerPoint format. If you click on the orange link on that document, it will take you to the web page for the kit, and you'll see a link on there that has the more detailed directions that spell out how you can change the prep and the protocol for your students. So for example, in the CRISPR gene editing lab, each group typically works with 4 different samples, A, B, C, and D. If you need to do this in a socially distanced classroom, that might look like giving each student their own responsibility of a tube. So one student might get tube A, another student gets tube B, so on and so forth. Then at the end, they can compile all their results together and use each other's data to come up with their claims about gene editing. So that's just one example, but there's different ways to do this. And if you need further help adapting these, please don't hesitate to ask us. We'd be happy to help you out.